Hi, good morning, everyone. Good morning, good morning. I'm Donna Buss. I'm with the Science Outreach Center. Thank you for coming this morning to Tiger Talks. We're thrilled to have you here. I am so excited to welcome Dr. Leah Casabianca this morning. She is going to talk to you today about pollution and plastics and the impact that it has on our environment and our world. So I'm going to turn it over. Oh, I wanted to tell you, feel free, use the Q&A little section. I always forget to tell people to do this. If you want to put information in or ask questions, use that Q&A button that's at the bottom of your screen, and you can simply just type in there, and I will be able to answer those questions or get Dr. Casabianca to answer the questions. Brilliant. So at the end, we'll save about 10 minutes of our 30-minute time frame, and we will have question time. But I'm going to turn it over to Dr. Casabianca. Thank you for being here.
Great. Thanks, Donna, for that great introduction. Thank you, everyone, for joining this morning. I'm going to talk to you about why small plastic pieces are a big problem when it comes to plastic pollution. We all know that pollution is a bad thing. We want to try to recycle our plastics or at least get them into garbage or into the landfills. But unfortunately, plastic does find its way into lakes, rivers, oceans. This was extremely important during the pandemic when we had a lot more takeout. So there's a lot more styrofoam, a lot more plastic forks, a lot more PPE, these masks, really increased plastic pollution. And again, whether we like it or not, a lot of plastic ends up in an ocean. So if you think about a piece of plastic in an ocean, it's exposed to high salt concentrations. You've got salt water. There are waves, so it's kind of, breaking the plastic apart. There's light from the sun that's going to lead to breakdown of plastic. So a large piece of plastic pollution eventually gets broken down into smaller and smaller pieces. And when it gets very small, we say that this is micro or nanoscale plastic. And when it gets to this size, it's about the same size as fish food. So fish will think that this plastic is their food, and they will eat the plastic. And then the bigger fish eat the smaller fish, and we eventually eat the bigger fish. So we are ingesting some plastic through the food chain. We could also be injecting this micro and nanoplastic just from the things that we eat. So if you seep tea in a plastic tea bag, this releases billions of micro and nanoplastics into your tea. So we're getting this plastic in our body, and this is becoming a main concern. A lot of people are worried about the amount of nano and microscale plastic that we are ingesting and is getting into our bodies every day. However, you have to think about a piece of plastic in a polluted waterway. In this polluted water, there's also other molecules that are not so great to be eating. So there may be pesticides, there may be herbicides, there may be pharmaceuticals. There's a lot of other nasty stuff in a polluted water. And the thing about these micro and nanoscale plastic is they tend to absorb these other toxic molecules. So if you have a fish that has some plastic nanoparticles in its tissue or in the body, and we eat the fish, or eventually this plastic nanoparticle, if it settles in a fatty tissue, it could potentially then release these toxic chemicals that it's absorbed from the polluted water. So we have to worry about not only ingesting plastic, but we have to think about all these other toxic molecules that we may also be ingesting at the same time. And the reason why micro and nanoscale plastic is really important is because, as we'll see in a little bit, the smaller the particle is, the more surface area it has. And the more surface area, the more of these small molecules are able to absorb onto the surface. So one of the things we're looking at in my lab is if you've got this molecule, so this is the chemical structure of a pollutant, and let's say this white sphere is a plastic nanoparticle. How does it absorb? Does it absorb on the side like this? Does this end go towards the plastic nanoparticle? Does this end go towards the particle? So if we can understand these kinds of things, we can understand more clearly how things bind. And then we can start to maybe think about how to prevent these small molecules from binding. And also we can look at if there are certain molecules that are toxic that we know absorb onto plastic nanoparticles. Maybe we need to screen our fish for those molecules in particular. So this is what we're working on in my lab. But first, I wanted to answer the question, do smaller particles really have a larger surface area? So is this small scale, nanoscale, and microplastic, is it really much more dangerous than large plastic? So to do this, I have here a piece of, let's say, plastic pollution. So this is a block of styrofoam. So if we look at it, and let's say I want to cut it into 9, make 2 cuts here and 2 cuts here. So we can say we have 9 squares here on each face. And a cube is going to have 6 faces. So it has one here, one here, one here, one here, one here, and one here. So if we think about the surface area of this cube, it's going to have a surface area of 9 times the 6 faces, so 54 of these units. Now, if I cut this large plastic particle into all of the pieces, right, we can count them. There are 27 of these smaller particles now. And each of the particles has a surface area of, instead of 9 now, it's just 1. But again, it has 6 faces. So we now have 27 times 6. So the surface area of these 27 pieces is now going to be 162 units instead of 54. So we can see that when we start to cut a big piece of plastic pollution into smaller pieces, we increase the surface area. And if we take each of these little squares and cut them again into 9 squares, we go from 162 to 486. So the surface area increases dramatically when we start to get to smaller and smaller particles. So to examine this again, we're going to do an experiment. So we're going to think about M&M candies. So if we have the same mass, so the same amount or the same chocolate of regular and mini M&Ms, and we want to know which will have more candy coating. So the candy coating is going to be on the surface. So the one with the larger amount of candy coating will have more surface area. So in science, we make a hypothesis. So I want you to help me with this hypothesis. So there are 3 possibilities. The regular M&Ms may have more candy coating. The mini M&Ms may have more candy coating. Or they might both have the same amount of candy coating. So you can put in the Q&A your guess. So either A, B, or C. So put that in the Q&A, or if you just want to think in your own head, choose what you think is your hypothesis. Do you think that the regular M&Ms have more candy coating, the mini, or that they both have the same? I'll give you a few seconds to think about it.
You can simply type in A, B, or C. That'd be easy.
Right.
Yep, there's an A. Keep coming, keep them coming. A, A, A. Any more?
Going.
Another A.
All right, sounds like we have a lot of A's. So let's go ahead and do the experiment.
Oh, somebody's picked C.
Okay, let's see.
Go, Oliver. Getting out there.
Great, good answers. We'll do the experiment now to test our hypothesis. So I weighed out different masses of regular and mini M&Ms. So this is about 1 gram of the regular, 2 grams, and 3 grams. And these are the minis. So about 1 gram, 2 grams, and 3 grams. So they were about this mass, but I recorded the actual mass as well. So then what I did to figure out how much candy coating they have, I added water and sort of washed off the candy coating. So you can see here, I tried to do it pretty quickly, so that all the candy coating has dissolved, but not dissolve any of the chocolate. So I waited until the M&Ms were white. And then what I want to do is, you see that the color washes off. So I looked at blue and orange M&Ms. The color washes off pretty easily. But then if we want to know how much candy coating was there, is related to the concentration of the orange or the blue molecule. So we need a way to, once we've washed this off and taken the water, we need a way to examine how much of this blue or orange molecule is in the water. That kind of tells us sort of a measure of how dark this is. So how blue is it and how orange it is. So to do that, we use something called UV visible spectroscopy. So this is the instrument that we used. This is my student Rajan using the instrument, putting a sample in. And this is the result that we get. So we get something called a spectrum. And this is measuring the absorbance at different wavelengths of light. So we see that the blue M&M absorbs around 630 nanometers, and the orange M&M absorbs around 480 nanometers. We can see that the orange has a higher absorbance. The blue M&M has a lower absorbance. So that's related to the different molecules that make the blue or orange color. And what's interesting is if we put the spectrum of visible light underneath here, we see that the blue M&M absorbs in the region where we see orange light. So the blue M&M is absorbing the orange light, and that's why it appears blue, because we know blue and orange are opposite colors on the color wheel. And then the orange M&M absorbs in the region of blue light. So it's absorbing the blue light, and that means it appears orange. So we can see where in the visible spectrum each of these molecules absorb. We can also use it, again, this absorbance, so how high the peak is. We can use that as a measure of sort of how dark the water is. So how blue is it or how orange is it? So we did this for our 3 samples of the regular and mini M&Ms for both the orange and blue M&Ms. And so for the different masses, we can see the absorbance. So we see for the regular M&M, this is the absorbance, it's the straight line. For the blue regular, it's here. But for the mini, when the mass is the same, the mini M&M actually has a stronger absorbance. So that means that there's more color in the mini M&Ms than in the regular. So we can say that in conclusion, for the same mass of M&Ms, the mini M&Ms actually have more candy coating than the regular M&Ms. So this makes sense because we've said that smaller particles have a larger surface area. So if we take the same mass of mini M&Ms, we're going to have more surface area and more candy coating. So we've said that we've used UV visible spectroscopy to do our M&M experiment. But I wanted to kind of change gears and just talk a little bit about what we do in my lab. So we use something similar. It's also spectroscopy, but it's called nuclear magnetic resonance spectroscopy. And it uses a big magnet. So this is my student Emily standing next to the NMR magnet. And this is the instrument that we use every day in my lab. This is what our sample looks like. We put our liquid sample in a glass tube and we put it into the magnet. And these are what our results look like. So we get a spectrum similar to the UV-Vis spectrum, but it gives us different information. And this can tell us information about the molecule that we are looking at. So how do we use this to understand how small molecules interact with plastic particles? So one of the things we can do is we can determine whether a molecule binds to the plastic or not. So we looked at this molecule. This is the chemical structure of something called perfluorooctanoic acid, so PFOA. And this is what we call a NOSI spectrum of this molecule by itself. And you'll notice we have some red dots. So by itself, it has red dots. When we add the polystyrene nanoparticles, so these are nanoparticles composed of polystyrene, so essentially styrofoam, but they're very, very small particles. When we add that to our PFOA, we see that instead of red circles, we have now blue. So the difference in color between the red and blue, and you also notice that here there was no peak, no circle. Here we have blue circles everywhere. That tells us that this molecule is binding to the plastic nanoparticle. So when there's no nanoparticle, there's no binding. when there is a nanoparticle, there's binding between this molecule and the nanoparticle. So my students, Emily and Sakina, did that work. Another thing that we can use NMR to determine is what part of the molecule binds to the plastic nanoparticle. So this was done by my student Yunji. We took another similar spectrum, and based on this graph, we can tell this molecule, this is the chemical structure of tryptophan. So it's in Turkey. It's what makes you very sleepy after Thanksgiving. But we looked at this molecule, and we saw that it also binds to plastic nanoparticles. And based on this graph, we saw that this end of the tryptophan molecule was closer to the surface of the nanoparticle when it was bound than this end. And this end is farther away. So we can use NMR spectroscopy to determine what part of the molecule binds to the plastic nanoparticle. And then the last thing that we can do is we can look at competition. So if we have more than one molecule, we can see which one is going to better bind and which one may outcompete another molecule for binding sites on the nanoparticle surface. So these are 3 antibiotics. So antibiotics might also be present in polluted waterways along with plastic pollution. You've maybe taken amoxicillin if you've gotten sick and needed to take antibiotics. So you may recognize amoxicillin. Metronidazole and levofloxacin are usually used for veterinary uses. So if you live on a farm, if you have animals, that you may have given them metronidazole or levofloxacin. But we looked at amoxicillin and metronidazole bound to plastic nanoparticles. And these are the graphs that we got that indicate binding. When we add amoxicillin and metronidazole in the same sample, the peaks for amoxicillin look the same, and the peaks for metronidazole look the same. So amoxicillin and metronidazole don't compete with each other for binding to plastic nanoparticles. But then when we added levofloxacin, you can see that the peaks for amoxicillin went down. So this indicates that the binding is decreasing. And the same thing happens for metronidazole. So it's up here and now it's down here. So when we add levofloxacin, that makes the binding between amoxicillin and the nanoparticles or metronidazole and the nanoparticles less. So the levofloxacin is outcompeting the other 2 antibiotics for these binding sites. So we can look at things like this, like competing molecules, which one can outcompete the other for binding to the nanoparticle surface. So with that, I would like to thank the members of my group and the funding, and thank you for your attention. I'm happy to take any questions.
Yeah. Fascinating, fascinating how we can maybe clean it up a little. All right, questions and answers. Come on, throw in your questions in that Q&A little box at the bottom, and we'll be happy to try to answer them, whatever they might be. Okay, somebody lives on a farm, but they don't know what they give to the animals, so now they know. So they're super cool.
Yeah. So one of my sisters, actually, Anna, her dad is a veterinary tech. So she actually tells me a lot about the different diseases that animals can have and problems that animals can have. And it was really interesting talking with her. Yeah.
Other questions you might have about pollution, plastics, the ocean? Dr. Casabianca, tell me a little bit about what made you get interested in this. How did you figure this out? How did you get to this place?
That is a very good question. So I think it was 3rd grade when we started doing chemistry experiments. I don't know if we called it chemistry, we maybe just called it science. But yeah, I really liked mixing things, and we did a really neat experiment. I remember where we're trying to determine if you have salt or sugar. So if you look at it, salt and sugar looks the same. It's chemistry, so you're not supposed to taste it, but how do you tell if it's salt or sugar? And so one of the things you could do is heat it. And so the sugar will melt, but the salt will not. So that really got me interested in chemistry and how do we use chemical techniques to identify something or to see how things are going to interact with each other on a molecular level. Yeah, so I think it was third grade that I got
Very
interested in
cool.
ocean. Yeah.
All right, so one question is, a couple questions came in. So one is, does pollution hurt all animals?
I think so. Yeah, unfortunately, because everything is so interconnected. So again, even if it's a small fish that's going to eat the plastic, there's a lot of complications to just that small fish, right? It could eat the plastic instead of food, so it doesn't get proper nutrition, so it could starve. Um, the larger pieces, bigger fish could eat it and they could choke on the plastic or they could get entangled in it. But even, yeah, if the small fish starve and then now there's no food for the larger fish. So yeah, everything is interconnected. So pollution hurts everyone. Yeah. Good question.
And we're not talking about just ocean, we're talking about land. It could happen. Yeah.
Same, yeah, yep. Land, soil, even rivers, lakes.
Mm.
Yeah.
All right, with your research, what kind of solutions are you all coming up with to decrease this pollution, not population? Are you focusing
Yes.
on freshwater or saltwater?
So one of the things that we're looking at, so I've talked about plastic as sort of a negative, plastic nanoparticles are pollution, but we could turn this into a positive. So we could actually design plastic that deliberately absorbs certain molecules.
Mhm.
So these fluorinated molecules, these are something that people are concerned with a lot lately because they're everywhere. They kind of come from like Teflon and firefighting foams. So if we can design plastic that does absorb these molecules, maybe we could use the plastic to kind of clean things up. So if you think about taking plastic nanoparticles and putting them into the ocean in something where they're not going to disperse, maybe not a bag, but somehow but we've got these plastic nanoparticles that have a lot of surface area. If we put them into the ocean, we can absorb all of that perfluorinated compound and then remove it in a net or somehow remove those plastic nanoparticles. That could be a way to improve cleanup. So yeah, we could kind of take it as a negative, but we could also turn it around into a positive.
Yeah. And these next two questions, they kind of do that. So I'm going to kind of lump them together.
Okay.
One of the questions is, can you melt the microplastics in the sea together and make one big clump so you can take it back to land and use it for other things? And then the other question that kind of is on that is, why doesn't someone, let's go with that, just create a machine that could suck up this pollution?
Yeah, I mean, there's lots of people are working on a lot of these solutions, and those are really good suggestions too. Yeah, to kind of could we condense all of the small plastic and then collect it and then just take it out? That's a really good solution. So I think that whoever asked that, you're potentially going to be a future engineer and a future
Mhm.
scientist worker that's going to contribute to these solutions and develop these solutions to help clean up the waterways. So that's amazing. Yeah.
Yep. So scientists are currently working. They're working as hard as they can. Okay.
Yeah, and there are many different solutions, and I think it's going to take all the different solutions, all the different ideas to really, because I think just one solution is not going to do it. We need to number one, reduce waste. So we need to figure out how we can avoid single-use plastics, as well as improve recycling, and then improve cleanup. So we need to work on it from all fronts.
Yeah.
Yeah.
Is there an animal that exists that eats plastic on purpose to live?
So there are people are developing or discovering like worms that will eat plastic and degrade plastic. So yes, there are organisms that can be used to get rid of the plastic. And so that's another solution, another way that some things that people are working on. Kind of outside of my area, but yeah, again, we need all the different solutions. So.
Do you only study water pollution?
Great question. So far we've only looked at water because our instrument that we have can only look at things in solution right now. But we actually it just arrived yesterday, an instrument that can study things in the solids. So we're going to look at soil and things like that in the future. So yeah, very good question.
How many animals eat plastic a day? Ooh, that's a big question.
That is a very good question. I have no idea, actually. Um, that's a very good question.
A lot, because there's a
Yeah,
lot of plastic,
a
right?
lot. Yeah. I mean, and even just the fish, just constantly, yeah.
So Oliver, digging Oliver, he says, give me the money in a vacuum and all the plastic will disappear. Love it, Oliver. Any other questions? Ooh, what has been your favorite experiment so far?
Ooh, I like this one that I'm showing right here, the transfer nosy, because it's just so amazing. You see nothing and then boom, everywhere. So it's really nice. You can see it doesn't bind, binds.
Yeah.
Yeah, and it was fun to set it up. So
Did you think that was going to happen when you did your hypothesis?
um, no, we were trying yeah, this happens in science a lot. We were trying to do something else. So we were trying to use this no-see experiment to see different things. So usually this experiment is used to see distances between two atoms in a molecule. So we were trying to use it to see distances. And what we saw was, we were trying to see just a few of these peaks, and we saw, oh my gosh, they're everywhere. And so then we started to look into, well, okay, why does this happen, and how can we use it? So
Oh,
yeah,
yes.
it happens a lot in science. You are trying to do one thing and you see unexpected results.
Right.
And a lot of times that's the most exciting.
Yeah. Any idea how much plastic is actually sent to the ocean?
A lot. A lot, yeah. Um, too much.
Too much.
Too much.
Is there a way to recycle microplastics?
I think that there should be, but the problem is that, yeah, they're so small. So it's hard, again, to kind of collect them. And then it's even a lot of times hard to detect them in water. Another issue is that the surface is kind of unknown. So what molecules are on the surface, what kind of atoms are on the surface, a lot of it is still being explored or being understood. Because you have all of these issues in the water. You have the salt, you have the waves, you have the sun. So it's not really known what's on the surface, and that makes it hard to detect nano and microscale plastic.
Yeah, the constant change, the constant movement.
Yep.
Yeah,
Yeah.
not
And
like you
so,
would have a terrarium or aquarium. So
yeah, right. And then the light from the sun that can initiate reactions that are unknown. And so other people who are in a similar field but not my field are looking at, yeah, what actually happens to the surface of plastic? How does it break down? What's on the surface? And looking at ways to how do we detect it? But yeah, if we could detect and collect it, we could somehow recycle, again, remove it and recycle it. But there are also challenges even to just finding it. And I think it's not even really known how much microplastic is out there. So
so we recently looked at somebody saying this. We recently looked at pond water and we got to see live organisms. So they're asked the question is, what do nanoplastics look like under a microscope compared to living organisms?
yeah, so they would look like well, they wouldn't be moving. They would kind of look like a clump. Um, and yeah, the size, microns or even smaller. Um, yeah, they just kind of be a small clump, and then you kind of envision them as being spheres, but right, they're not always going to be spherical. They're going to have different shapes. They're going to have different sizes. So that might be a good idea for maybe the next Tiger Talk. Maybe I should include a picture of what these look like under a microscope. That'd be really nice.
Why do the fish eat the plastic?
Mhm. So yeah, the fish, they can't tell that it's not food. And they may be just, taking the water through. Oh, I don't know fish biology that well, but they're just kind of taking in the water. And the same thing with, why do we eat? If we go here, yeah, why do we eat the plastic? Well, it's in our tea, so we can't help ingesting it. So yeah, it's a little bit of accidental injection that's kind of unavoidable. Yeah. Yeah. Good question.
What other kind of projects do you do every day? Like it,
Mm. So I teach. So I have classes where I teach students. I do things like this. I do outreach. I do Tiger Talks and things like that. Do a lot of budget stuff. That's not fun. A lot of paperwork, but that's just part of life. I train students. I work with students in the lab. So my, let's see if and back to my acknowledgement slide. So these are all students who work in my lab. So I get to work with them every day, which is wonderful. They are great. They're amazing. So we do experiments together. So I kind of teach them how to do the experiments, and they teach me a lot too. So they, teach me what they know. We work together. So most of my day is really spent doing experiments with the students.
When did this all start? How was this turning into a problem? How did it start turning into, how long ago?
Yeah. Well, the nice thing about plastic is it's great. It's cheap. It's easy to make. Making a plastic fork is cheap, and then you can just have the fork, you can eat, and then you can throw it away. It's very convenient. So yeah, once plastic was invented and people started to realize, hey, this is great. They started using it again and again and again. And it's great because it's safe in terms of healthcare, right? So things that you inject, a plastic tube, if you want to inject it, that that's clean. It hasn't been used before by anyone else. And the same thing if you have a plastic water bottle, it should be clean. It's not going to have any contamination. So plastic is really great for a lot of things. It's really cheap. It's really safe. Even, plastic packaging, right? So your food doesn't get dirty. That's a great thing. But yeah, as people started using it more and more, that's when we started to not be able to kind of control where it goes. And, we do try to dispose of it properly, right? We try to recycle. But yeah, I think it's going to take, let's try to use less plastic. Let's try to recycle what we use. And then let's try to figure out, what we can do at what we call the end of life. So after we've used the product, whether it's a fork or a container or something, what do we do with it then?
Right. I do value your time, and I know that we're at the top of the hour. I do want to let you know that our next Tiger Talks is going to be Thursday, October the 16th, and we have Dr. Naveen Vecha, and he is coming from none other than NASA, people. So tell your family and friends to sign up. I'll send out the registration links probably tomorrow, and then we'll put it out on Facebook. We'll also be posting this on YouTube, so I'll send all that information out by email. He'll be talking about his career journey with NASA, and I want to thank you all so much for attending. Thank you for your time. Dr. Casabianca, thank you so much for your time. And
thank you everyone for your great questions.
yeah, thanks for those questions too. I hope you all have a great rest of your week. Anyway, take care, everybody. See you next month.